So my name is Andrea, and I am a fifth-year graduate student. And this is a relatively new project, but I thought it was a really great opportunity to talk to you all about what is brain development, what do we know about typical brain development in children and adolescents, and what do we know and want to know about brain development in 22q. So anybody who has watched, themselves as grown up and children, that development is a continual process, that every day we go through these small steps, it's incremental, that you can't always see these changes, but they are occurring. When we invite your child into our studies and we do an imaging scan, it's really taking a snapshot of what their brain is at that point, and we make inferences about the development prior to that scan and about the future development. But it's only one snapshot. As much as we would love to be able to continually study brain development, it's not feasible, right? Luckily, what we hope to do, and some studies have done, is done a longitudinal study where they bring back children and study them a couple times. So there's a study that was done in UCLA, Heather showed this data before, where children came back every 2 years, and we were able to actually see changes in brain maturation. So what we're looking at here is a picture of the whole brain, and the colors indicate the gray matter volume. And so red here means a lot of volume, and purple means less volume. Okay, and that's not a good or bad thing. What you can see is you compare the 5-year-old brain to the 20-year-old brain, there's kind of two major points. One is the whole brain isn't one color. So throughout the brain you have different volumes, and that's typical. And then over time there's some regions that change more and there's regions that change less. There's this little pocket right here that's red at 5 and it's still red at 20, versus there's regions like here in the prefrontal cortex that are red at 5 and will become purple at 20. So it's a very dynamic process. What we really want to know is what's the trajectory, what's the rate of this change over time. And a trend that we see in development is that it's not linear. We can all think of things that increase and then decrease. If you think about your body's metabolism, when you're a kid, you eat a lot, and then you get older, and maybe you don't eat as much and process it as fast. You get this rise, it's called an inverted U shape. So this process of developing, of getting an increase to a peak, and then a steady decrease is a normal function of brain development. So whether or not we're looking at total volume of the brain, you get this increase, peak, decrease, or particular regions, you get this shape. What's interesting I want to note, though, is there's a lot of variability in which this happens. So here you see two different lines, and the blue here is for males, and the red is for females. You can see there's already differences in gender. The shapes aren't different in the lines themselves very much, but there is an offset. What we don't really know at this point very well is to what extent does this pattern occur in children with 22q. Okay, so this is the pattern I just showed. What's the expected trajectory in 22q? So this is a typically developing pattern. That's in here, we're looking at cortical thickness. This is how much gray matter. Same occurs for volume. And you get this increase during early adolescence and the steady decrease in puberty. What we don't know, what we want to really find out is what's a pattern that occurs in 22q? Is it an atypical pattern, and how atypical is it? You could imagine there'd be a pattern where you get, there could maybe catch up to brain development, or maybe there is no change. This is what we aim to find in our studies here. So there's a little, but that's not to say that nothing is known. There are some studies that have looked at this. Most early studies looking at brain development found that overall brain size or brain volume, if you measure it as a whole, tends to be smaller in children with 22q. But then once studies started to look at specific regions, we actually found that, yes, there are some regions that are smaller compared to typically developing children, but there are also regions that are actually enlarged. So then the follow-up question is, how does that change as that child gets older? Do the larger ones continue to get larger, or do they get smaller, and what rate does that happen? So this is one study that was done looking at, this is a cross-sectional study, so they had children who were 6 to 9, 9 to 12, 12 to 15, and so on. And they were measuring cortical thickness in the whole left hemisphere in this case. And what they found is, these maps are actually showing you in blue regions that were larger in 22q. So you see that there are some regions that are larger in 22q and some regions that are smaller, even though overall there was more thickness in the younger kids compared to the typically developing controlled children. But by the time we were 18, there wasn't a measurable difference. So we're already seeing a difference in rate over time. They also did a longitudinal study where they had some children who came back multiple times, and they looked at, looking from 6 to 12-year-olds, they saw that some of these brain regions actually didn't have a lot of change. So we're already getting an idea that there are some changes that are going to occur in the brain, but it's not a global process. So we kind of want to follow this method, and the way that we're doing it is using a whole brain technique, a state-of-the-art algorithm that I'm very excited about, part of my dissertation project, that lets us kind of answer some of these questions. So the way that this works is a child comes into the lab, usually in that first time, and we get a brain scan, so we get an image of what their brain looked like. And then we've had 10 children at this point with 22q that have come back for a second visit. So what we have here, the 10 participants that come back, this first dot is the age they were when they first came into the lab, and then they usually come back about 3 years later, and then this is that second age, so you can see we have a nice span from childhood to early adolescence. So they came back a second time, we got a second image of their brain, and what this algorithm does, in a sense, to reflect the development that the brain went through from time 1 to time 2, is it reshapes that first brain to look like the second, and then it gives us a measurement of how much certain regions change. Did they get bigger? Did they get smaller? Did they move a little bit? And we can measure that. So another way to visualize that, imagine you had a four-door sedan, and you really wished you had a Volkswagen Beetle. This algorithm would reshape that sedan to look like a Beetle. And we can measure how much you had to change certain regions, right? The front shape here would have to change a lot more than the wheel barrel, where the wheel goes. I don't remember what that's called. So that's what this algorithm does. And so this algorithm, kind of in a sense, we're using that in a way to reflect the brain development, in a sense, how much maturation occurred at a particular region. So again, these are very preliminary results, but we just wanted to show you what we have. So these are kind of measurements, like I said, of cortical maturation. What we found is growth or increases in the sulci of the brain. So this image here is just a brain. I want to show you what a sulci is. So the brain has all these folds, and the sulci are basically these folds inside, the crevices of the brain. So this is the example data of 2 participants, so 2 of these children who come back. And the reds here indicate increases in size from time 1 to time 2. And you can see that for the most part, they're located within the sulci, within the gray matter. And what's interesting is that these tend to be, in the participants we've seen so far, to be in locations that have been implicated in some of the behavioral tasks we've done, like what we call the interparietal sulcus, and the frontal cortex, and that's really promising. That's where we would almost expect to see differences, because that's where we see behavioral changes over time. So this is exciting prospect of showing us that there are changes that are occurring, and we hope to measure them more systematically and quantitate those changes. And hopefully next year I'll have much more to tell you about this. So the idea is that we know that there's the changes that occur. What we don't know really is, what does that mean when you have a change, when you have an increase or a decrease? What's the appropriate size for anything to be? Is there a just right size? Is it too big or too small? And can you, given what we know about plasticity in the brain, is that something that's something that we should be able to change and target? So I really hope is that even though we don't know what the right size is, and given the extreme amount of variability that exists in the typical population as well as in children with 22q, can we use that to inform treatment options? So if we know that a region is maybe smaller than it ought to be for a typical development, if we know that early and we target treatment, can we then change the potential trajectory it's on and get it to where a more appropriate or ideal location with time? And so with that, I'd like to, of course, thank all of our participants' families, especially those who come back and continue to come back and help us. So thank you.